Right, so this is an essay question. It's coming from June paper of 2016. It says, describe the location, the structure, and the functions of the DNA molecule and the process whereby copies of this molecule are made. Okay, so if you look at this essay question, there's a lot of marks that's coming from the essay question. It's important that you read it carefully and you break it down thoroughly, right? So what's the verb firstly? Right? What's the action word that's telling you what to do in this specific essay? Right, good.
Describe.
Thank you, right? Good. Describe. So whatever comes after the word describe is now important for us. Sometimes there's a leading statement to say DNA is a crucial molecule in our bodies and a full stop. There's no question there. It's just guiding you to say this is the topic we're talking of, right? So good. Describe is the verb we're looking for. So now there's many different parts to this question. It says describe the location, structure, and functions of the DNA molecule and the process whereby copies of this molecule are made. So one of the steps or the ways to answer an essay completely would be now to give subheadings. So can someone give us the subheadings? What would the various subheadings be? So that we make sure we answer all aspects of the question.
The location of the DNA and the structure of the DNA and the functions of the DNA
Good,
molecule.
Kopano, right?
And then
So
the DNA.
you said the location of the DNA, the structure of the DNA, the functions of the DNA, and then here's the last part, and the process whereby it makes copies. So you can have 4 subheadings. It's not compulsory for you to have subheadings, but I suggest that you do because it makes sure that you answer all aspects of the question. Make sure that you also are sticking to the question that was asked, that you don't go off topic. So what's the location of DNA firstly? Let's start answering the question now. Right. DNA is found in the nucleus, good Katlego, right? There's one more place that we've also mentioned. Right, Avraj, you are correct also. Mitochondria, good, right? So if you look at the exam guideline, it says that DNA is found in 2 places. That's in the nucleus as well as in mitochondria. Right? It's also in the chloroplast of plants, but that's not in the exam guideline. Now the structure. Who can describe, remember it's not naming, it's describe. Now describe the structure of DNA. There's many parts to this here. There's a lot that we can speak about here under the structure of DNA. Okay, it's made up of nucleotides, that's correct, Katlejo, right? Okay, we can elaborate on those. Right, so remember, it's not name the structure. Naming would be there, part of that. But now, if you're saying it's made up of nucleotides, then let's describe the nucleotides. Let's go from there. Yeah, remember, thank you, Madam Lahej. We need short sentences. You don't need to type up your whole essay and send it. You can just send sentence by sentence, and we can just add to each other's answers as we go along. So we said let's build on there. Okay, Muaz is correct. Double-stranded helix. Right, that's a separate point from Katleho's. That it's a double-stranded. Right, it's one mark, and it's a helix again. That's another mark. And then within that, it's made up of nucleotides. What Katleho said, but I said let's build on from what she said. Right. Good. Abra says the nucleotides are made up of phosphate group, deoxyribose sugar, and one thing is missing there. Abrar, right? You said it's the phosphate, deoxyribose, and 4 nitrogenous bases. Good, perfect. Good, right? And what are those nitrogenous bases? Let's describe them. Right, so there's 3 parts that make up nucleotides: phosphate, the deoxyribose sugar. Very good, Abrar. Remember, if you just say sugar, you don't get the mark. If you're speaking of RNA, you'll say ribose sugar, and if you're speaking of DNA, you'll talk of a deoxyribose sugar. And then the 4 nitrogenous bases that we get are good thymine, adenine, cytosine, and guanine. Thank you, Katlejo. And then we had also someone else agreeing with that. Okay, right? So we've got different nucleotides, and then who can add? Good. Adenine bonds with thymine, good, and obviously cytosine joins with guanine. And how are good more? That's a complementary pairing we're speaking about, and you said they are bonded. I think we can also speak a bit more about how they are bonded. And how are the nucleotides bonded within a DNA molecule? Weak hydrogen bonds, perfect, good. Right. I think we probably have enough on the structure there. We'll just look at that essay, the memo. Right, so it says DNA is located in the nucleus, right? Or you could say chromosomes or within genes or in the mitochondria. They gave only one mark for that. It's a nucleic acid, right? We didn't say that. It falls under the group nucleic acid. There's two that we learn about, DNA, deoxyribonucleic acid, and RNA, which is ribonucleic acid. It is double-stranded, and it's a helix, right? So we said it's the double helix. That's the 2 marks. It consists of nucleotides, which have nitrogenous bases, 1 mark, phosphate, and deoxyribose sugar. Then to name them is another mark, and then to talk about the specific bonding A with T and C with G is another mark, and they are joined by hydrogen bonds, right? And then the last part was they asked also for the function of DNA. What are the functions of DNA? Carries hereditary information and protein synthesis, right? Good. I know what you're trying to say, but you'll have to just give a bit more on protein synthesis. That for protein synthesis, it has the code for protein synthesis, right? So sections of DNA carry the hereditary information. Replication, Katlego, you are correct. It does undergo replication, but they're not giving that as its function in this memo, right? Because I think replication was given in the question, if I'm not mistaken, right? So they're saying DNA contains coded information for protein synthesis. So it doesn't do protein synthesis, but it has the entire code for protein synthesis, where we know that during protein synthesis in transcription, it unwinds and the copy of the code is made in the form of mRNA as a complementary code. And that code is then taken to the ribosome, where translation will occur and the correct sequence of amino acids will take place to form the protein based on the DNA's code, right? So the role in protein synthesis is that it contains the code for which protein needs to be formed, right? So they said the last part was the process whereby copies of this molecule is made. Did they name the process? No. So what is the process whereby copies of the molecule is made?
DNA replication.
Good, right? DNA replication is the first thing to name it. And we wanted you to then describe it, right? So can you describe this process for us? Right, it occurs in the good Katlego, it occurs, right? So can you see, for just saying replication is 1 mark. Why, if something is not given in a question, and even if it's given in a question, there's no harm in writing it again. Replication, it occurs in the nucleus during interphase, good. And then the process is that the DNA double helix unwinds, correct? And then after unwinding, so it's untwisted, we find that it unzips, correct? And it unzips because the weak hydrogen bonds break. And then after that, what happens? Right, both strands act as templates. Very good, Katlego. That's what we need there. Not one strand. Remember, to differentiate this process of replication from transcription. Where transcription, 1 strand acts as a template. Here in replication, both strands act as templates. And now after that, what happens now to those strands? And so we can see the memo. The double helix unwinds, it unzips, the weak hydrogen bonds break. You should say that, you get a mark for that. Each strand acts as a template. Both of them are acting as template. Then nucleotides join complementarily to form new strands, right? That's correct. we're saying free nucleotides good, Moaz, and I'd be specific, right? I think this memo is not asking for you to be specific, but it's best to specify that DNA nucleotides, because once you say DNA nucleotides, you're automatically saying they've got a deoxyribose sugar and they've got the base thymine and not uracil. And when you're talking of the process of transcription, then you're going to say RNA nucleotides will join complementarily, right? So here, DNA nucleotides come and they join complementarily to either strand, and we get two strands of DNA forming. And you can add that it's controlled by, who can tell us what is this process controlled by? Okay, so free nucleotides, you had to mention free nucleotides, right? Good, Katlaho, it's controlled by enzymes. Right, so free nucleotides, 1 mark, build a new DNA strand on each of the original DNA strands, right? So you get two together by attaching their complementary nitrogenous bases. Or you could say A joins with a T and a C joins with a G, which means complementarily. And it results in two identical DNA molecules being formed, right? So that's important also, where in transcription, you're not getting something identical. You're getting one strand of mRNA, which is a completely different molecule. Here you're getting two identical DNA molecules being formed, and that's why it's called replication, making a copy. Okay, so that covers the paper. In this specific essay, the examiner decided not to give a mark for saying that enzymes are controlling this process, but you should say it because you don't know, a different memo might give a mark for that. Right? If we look quickly at the synthesis marks, there's 3 marks: R, L, and C. So R is for relevance, L is for logic, C is for comprehensiveness. You have to be on topic for R, nothing irrelevant, right? So if maybe you mention RNA and you're describing RNA in this process, you had DNA, but you also went into RNA, then you end up losing marks. So, or you start speaking about transcription, maybe you lose the mark for relevance. Logic is the information is given in a logical sequence, especially for replication, right? You need to have it in order, where you can't say, for example, the DNA unzips and then it unwinds. You're going to lose the marks in the content as well as for logic. And then comprehensiveness, did you answer the whole question, right? So for the location, structure, and function of DNA, you needed at least 7 out of the 10 out of 10 marks there, which was the maximum. And for DNA replication, you needed at least 4 out of 7 there to get that mark. Right, so that covers that topic.